Since the Human Genome Project was completed in 2003, there's been a huge revolution in nucleic acid technology, in that the cost of generating sequence data, and that's data for both DNA and RNA, has radically declined. And this means that we have a huge amount of data now accessible to us in a way that it has never been accessible before. And you can sequence DNA in humans, in many human populations or families. You can sequence the DNA of a tumour. You can sequence the DNA of all the bugs in your gut, of the bugs in the ocean, of animals, plants, and all of this can be used as basic data to try and learn more about what causes human disease, what helps the agricultural industry, or how the dynamic flora of our oceans is changing over time. So one of the things that the Institute for Life Sciences in their endeavour with this FioNA project has supported at the University of Southampton is our Next Generation Sequencing Annual Conference. We held our conference this May. It was the 5th year running, and we invite researchers from all around the Wessex region who use this next generation sequence data for their research. So we had people from the National Oceanographic Centre, we had people who work on chickens in collaboration with the Roslin Institute, we had lots of people from the Faculty of Medicine and the University Hospital Southampton who are working together in the Genomics England project, for which Southampton and Wessex holds one of the GMC, the Genome Medicine Centres, that's recruiting NHS patients to a national project looking at genomic data. We also had colleagues from Porton Down who've recently been to West Africa and sequenced the Ebola virus, coming and telling us about their research. So we were delighted that the IfLS was able to support this and support a student project to conduct sequencing and advanced research in this area. We're very lucky in that we've had research support from the KICRA charity, which is Crohn's in Children and Young People Research Association, and their funding has enabled us to accumulate these vast amounts of information, genomic information, on the children we've recruited to the Southampton Paediatric IBD genetic study. So one of the studies that we're using interdisciplinary skills for is a study of children with inflammatory bowel disease. So this is an autoimmune disease of the gut. It's similar to asthma in that it's not caused by a single gene, it's caused by a complex number of genes and environmental factors. And for each patient, a different set of genes can be causing the disease. So traditionally, these complex or common diseases have been very difficult to study. But now that we can use this next generation sequencing data, we have unprecedented access to a vast amount of genomic information to try and untangle exactly what is causing the disease on a case-by-case basis. So we work very closely with the Paediatric Gastroenterology Unit at University Hospital Southampton, and the entire consultancy team is on board, the surgeons, the research nurses, we're all involved in helping us recruit patients, so little children who come in and get diagnosed, and their families to this study. And we've just passed the 1,000 number of recruits to this study recently. For each of these children, what we try and do is extract some DNA from a small sample of blood that they provide while in clinic, and we analyse just the portion of the DNA that codes for genes and codes for proteins, and that's called the exome. So despite the fact that we're just looking at about 2% of the genetic information, we actually generate up to 25,000 points of variation for every individual. So this represents a very large amount of data. When you then combine this genomic data alongside a vast amount of clinical detail over time, such as body mass index, their health and well-being, what drugs they've taken, how well they've responded to them, age at diagnosis, and various histopathology reports, this then culminates to be a vast amount of data that can be very difficult to analyse. And so we are harnessing the local expertise in cutting-edge mathematical models and using machine learning to try and interpret those data in a more sensible way. And the aim is to try and identify the complexity of both genetics and other factors that are giving rise to this autoimmune disease in these children and their families. In terms of enterprise, FioNA is supporting this project at the moment by helping us chat to some consultants outside the university to get a different scope on the research potential and the wider enterprise potential of this project. So we generate big data. Big data is of interest to large computing companies that we're about to approach. Obviously, targeting therapeutics towards personalised diagnostics is a very large area, so we're also engaging with some pharmaceutical companies and multinationals in that regard. But we're also very interested in accruing day-to-day electronic healthcare data on the children in this study. It's always traditionally very difficult to collect accurate nutritional information over a long period of time on anybody, and we'd really like to collect this on children. So the IfLS and the FioNA project is helping us engage with individuals and putting us in touch with people who would be more expertise in that area of electronic healthcare data accrual, so that we can help enrich the data and of course then use our mathematical models to analyse those data in conjunction with the genomic data to further develop and push forward the boundaries and the capabilities of this technology. Ultimately, our aim is to try and get very personalised diagnostics on children, understand the molecular biology and biochemistry of specific autoimmune diseases, but basically be able to target therapies towards an individual set of susceptibility variants and environmental factors, such that patients can be given drugs that will really target their specific disease and therefore make them better quicker. In the case of these children, get them back to school quicker, back with their friends, back playing and having normal energy levels. But of course, in a wider interest, we would be able to expand this research to look at adult disease, but also to look at the vast number of other autoimmune diseases such as asthma, type 2 diabetes, arthritis, and try and use the same methods to try and understand those diseases and apply our learning in this set of children with inflammatory bowel diseases to a wider group of autoimmune disease.